All right, we're moving on to Unit 3, Biological Bases of Behavior. Remember, everything that you do psychologically is simultaneously biological. In fact, the DSM-5 recognizes that in all the mental disorders assume some kind of physiological or biological misfunction. Okay, so it's a very important unit to understand psychology. It's also very important because it's weighted heavily on your AP exam. We'll look at the makeup of your brain, and we'll look at these little guys, the little neurons that you have, and how they generate electricity and pass those messages on to their little friends. I got two of them, and how the chemical messengers send those messages and the pathways and how it might affect you. We'll also look at the anatomy of the brain, and we'll look at your central nervous system and how your brain communicates with the body. Okay, and we'll definitely spend some time looking at the techniques that we use now to look at your brain. We're learning so much about your brain every day. We know so much, but there's so much more to know. But right now, you are a brain studying a brain. So let's get started on biological bases of behavior. Okay, we're going to start our venture into biological bases of behavior. So let's begin by looking at the learning objectives. Explaining why psychologists are concerned with human biology, describe parts of the neuron, describe how nerve cells communicate with other nerve cells, and how neurotransmitters influence behavior, and how drugs and other chemicals affect neurotransmitters. Okay, so that's what we're going to be looking at. Okay, so originally, the first guy really to put together that different parts of your brain may do different things, like they specialize in certain things, was a guy named Franz Gall. So Franz Gall, this was a long time ago, and he was actually a very smart man, and he was attending his college classes, and he thought, there's people that aren't as smart as me that are getting better grades in here, and he wondered why. So he looked at them, and he noticed they all had like bulgy eyes, the guys that were beating him. So he thought, what was it about this? Oh, they must have better memories than me. So the part of their brain that has to do with memory must be pushing on the back of their eyes to make them all bulgy. Okay, so he came up with that idea. Then he looked at other people, their characteristics, and looked at bumps on their head. And he measured these bumps and correlated them with these people's ability, and he came up with what's called a phrenology skull. So this is what a phrenology skull kind of looks like. This is a model of one. I think he's very handsome. Hello, Mr. McConnell. I like him because he kind of looks like me a little bit. Anyhow, I digress. Okay, so biological psychology has everything to do with everything biological about you and how it affects our behavior and our mental processes, obviously. As was mentioned in the introduction, everything we do psychologically is simultaneously biological. There's something that changes in your brain to make you feel an emotion. There's some changes in your brain and in your nervous system that make you move muscles. There is all kinds of stuff going on. The other part with biological psychology we look at too is genetics, genes, and how they affect us. The gene and environment interaction as well. And we are biopsychosocial systems, right? We are biological, we have psychological aspects, and there are social aspects. Remember, these are the three main levels of analysis in psychology. So let's have a look at our neuron, which is the basic unit of our nervous system. Remember this guy? Okay, so this guy is depicted in this diagram down here. And the neuron are cells that run all through your brain. They're where the real action happens. There are other brain cells, which we'll talk about later, but the neuron is where all of the messages are sent and communicated through your brain. Okay, so in the neuron, you need to know the parts to begin with. Okay, we have cell body, dendrite, axon, myelin sheath, and we'll talk about multiple sclerosis when we go through this. But let's look at this diagram again. So here we have a motor neuron. What motor means is this is a neuron that will send messages away from your brain, okay, and to other parts of the system. They send messages away. Okay, so inside your brain, what happens is there's an amount of stimulation either from another neuron that's passed on to this neuron, which receives that information, or it's sent from a receptor, and it's received the messages in these spindly little things on the outside, which are called dendrites. Okay, these dendrites are what pick up the signals to tell this neuron whether or not to do its job or not. Where the job is actually created, which is an electrical impulse which will flow down the neuron, is in the axon. The axon is this blue structure that runs underneath the white encasing in this diagram and all the way to the end. Okay, so it passes its message away from the cell body to the other neurons, muscles, or glands. Now the white coating that you see that kind of makes it look like a sausage link, sorta, is called a myelin sheath. The myelin sheath is really important because what it does is help insulate that axon, because it is electricity that's passing through it, and this makes the electricity run more efficiently through the neuron. This is not in your notes, but you might want to write this down because you might need to know this. These little joints where there's a little space here, these are called nodes of Ranvier. And I'll put a little thing up to show you how to spell it. And they're there, those little breaks, because it may deplete the strength of the firing of the neuron if these are not in place. Now, the myelin sheath speeds it up. Now, you guys are at an age where your brain is starting to get fully developed, and what's happening inside your frontal lobes is myelination of your neurons is occurring. And so you're gaining more myelin on these neurons, so that you become more efficient and you'll be able to meet your full capacity of your brain. We have had research that suggests your brain actually continues developing up into your 40s. We used to believe it was 24. So the most of it happens by 24, give or take, but it still seems to develop even later in life. Okay, so the other thing with the myelin sheath, to relate it to you, a thing, multiple sclerosis. This is a disease maybe some of you know about, where it affects a person's ability to control their muscle movements and stuff. It's a very debilitating disorder, and it's usually degenerative, which means that it will get worse as you go. So what it means is multiple sclerosis means sclerosis is the hardening and lesioning of the myelin sheath. So as this myelin sheath gets hard and brittle and kind of breaks apart, lesioning is damage, the electrical signals are not passed through in a proper way, which causes the problems in multiple sclerosis. Okay, moving on. So we know this is the front of the neuron. When this neural impulse of electricity, it will flow through the neuron in this direction. Okay, and flow may not be the proper word for this, but it will be passed in this direction. And I'll explain why when we explain how these charges occur. Now, at the end of the axon, we have the axon terminal branches. So terminal means the end. This is the end of the neuron. Like a terminal disease or a bus terminal, it's the last stop. It's the end. Okay, and inside these end bulbs of the terminal branches is where our neurotransmitters, which are the chemicals that will send messages to the next neuron, because our electricity that's flowing through here cannot jump through a little gap that we have. Okay, back up in the top here, this area here is the cell body. We have a nucleus in the cell body. The cell body is the life support center. It controls the cell's growth. It controls the cell's health. Okay, so make sure you know those and the jobs of each of these parts of a neuron. So this neural impulse that goes down the neuron, the speeds can be incredible. They range from 3 kilometers per hour to about 290 kilometers per hour, which is really fast when you think it only has to travel the 6 feet of you or the 4'5" of you or however tall you are. And it's measured in milliseconds that it's so quick, which is 1,000th of a second. So we'll have a look. That neural impulse, we actually call it the action potential. So the action potential is the spark of electricity that will be passed down the axon. The resting potential of a cell means that it's ready to fire. And basically what it is that neuron, there are ions in and surrounding the axon, and ions are are electrically charged particles. But inside the neuron, or the axon, it is mostly negatively charged at this point, and it's mostly positively charged on the outside. In nature, everything wants to kind of balance out. It's like homeostasis. Everything wants to stay the same, and it will go to great lengths to stay the same. The outer casing of the axon, this is an axon of a neuron, by the way, in case you haven't figured that out, a blow up of it, there are little areas in the membrane which are permeable, which means these ions can pass through them. Okay, when it's in a state where it's mostly negative and mostly positive on the outside, we say they are polarized. Polarized meaning the opposites, right? We have positive and negative. They are separated. Just you think of like a magnet. You have a north and a south. One's like positive, one's like negative. And if you put them together, they don't. The north poles, they don't want to stay together. They repel. And when you put the north to the south, they want to stick together because they try to create homeostasis. They attract one another. Okay, so the membrane of the axon is selectively permeable, meaning it will allow things in and out of it. So when this happens, when these ions move, that's actually what electricity is. That's what causes the spark. And it depolarizes in this case. Once it depolarizes, there's a spark created. And then there is a refractory period. The refractory period is when there are sodium-potassium pumps, which is important to know, that will move the ions in and out of the axon, sodium, potassium being the main ions that are used. They're both the positive ions. Okay, there is excitatory versus inhibitory effects. So it's basically there has to be more positive than there is negative because the negative ones will inhibit it. The positive ones, when they move across, they excite that axon to create that action potential. Okay, so if we look at this diagram, we're looking at a blow up of an area of an axon and looking at it through the inside of it. This is the semi-permeable membrane that we were talking about. So once stimulation has occurred because there's a message sent from another neuron or from a receptor, it stimulates the neuron for this to happen. The sodium-potassium pump will move, the positive neurons will move into it, which depolarizes the cell, causing the electrical charge. Okay, and then the sodium-potassium pump, will remove those. So the electrical charge has occurred right here, but what it does is stimulate another membrane to open, and then more positive ions are moved in here as these positive ones are being moved out. So the spark, the electrical charge, basically one goes here, which stimulates the next, which stimulates the next, which stimulates the next. So it's not like a flow like water, it's one right after the other, like dominoes. Okay, so as it goes down the axon, this is actually preparing itself to fire at another time. But it needs to be totally back polarized again with more negative on the inside and positive on the outside before it is ready to fire. So when we achieve that point where there's enough of this flow of ions, it's called the threshold, when it actually will make that neuron fire. If you think of a rifle, the trigger of a rifle, it's a location basically. If you pull the trigger back slowly and you find that one little spot, if you go past there, the rifle fires. So that's like the threshold of the trigger. Same thing with a neuron. It reaches a threshold where it will start to fire. The all or none response, it means that the neuron fires or it doesn't fire, which means it fires exactly the same way each time. It doesn't change. It doesn't become more intense. It doesn't go faster. It doesn't do any of those things. It's the same, all or none. Again, same thing with the rifle. If you pull the trigger slowly, the bullet still comes out just as fast as if you pulled the trigger quickly. And it works very much the same way. Not with a trigger, though. So just one other thing on this. So how do we experience different sensations? For example, if we touch a desk very lightly, we can barely feel it. But if we pound our finger on a desk, it'll actually be painful. So why the different responses if the neurons fire exactly the same way? Well, what is probably happening is you are exciting more neurons, which send a different electrical code, different electrical message to your brain to tell you what you're experiencing. Okay. Or there's a higher frequency of firing. That neuron will fire more often. It won't fire faster, but it'll fire more often. So the synapse, this is at the end of the neuron, at the end terminals, okay, at the very end. there's a little gap between it and the receiving dendrites of the next neuron. That gap is called a synapse, or a synaptic gap, or a synaptic cleft. It means all the same thing. It's that little gap, and there's fluid flowing in there. And stored inside the vesicles, this would be the axon terminal of the sending neuron. This would be the dendrite, receiving site of the dendrite. There's little vesicles that contain neurotransmitters, which are chemicals that are passed between the two cells. So it will release it, they will float in there until they fit the receptor sites. And they work like keys. They will only fit in certain receptor sites. Now, this is very simplified because we're showing you one. Obviously, it's a very complicated thing where it happens in many places. There's usually more than one neurotransmitter present in the synapse. It just depends on where the threshold is met on the next one, which will cause the next one to fire or not. So what happens later is called reuptake. So if we look at this diagram, the action potentials, this is the synapse, and it's blown up here so you can get the idea. This is kind of what I've shown you, the synaptic gap, the neurotransmitters being released. But what happens once these are filled, it meets its threshold, and this neuron fires? What happens to the excess ones? Well, what happens is there's a process called reuptake. So the sending neuron goes, it sucks back the extra neurotransmitters in the cell, so that they're ready to be used another time when that cell is excited. They can be released again. There are certain drugs that work on this that will actually block these reuptake sections. For example, most antidepressants, which are SSRIs, selective serotonin reuptake inhibitors. It means if these neurotransmitters were serotonin, which is a kind of neurotransmitter that maybe you're familiar with, linked with mood. And so it's a depletion of serotonin is the theory there. So this neuron fires from the serotonin, which will send the pathway down a certain pathway in your brain, or the impulse down a pathway in your brain. The reuptake is blocked, so these remain in the synapse. So it's like this neuron has already sent more. So once this goes through its refractory period, is at its resting potential, these neurons are available, or neurotransmitters are available again to make this fire. So it's like you have more serotonin. You don't have more in this case, it's actually just more efficient because it doesn't get sucked back up. Okay, so that process again is called reuptake, where it just vacuums them back up. So some of the neurotransmitters that you need to know. We have acetylcholine, which is a very common neurotransmitter in your body. It enables muscle action by causing muscle contraction at its receptor sites near your muscles. It's involved also in learning and memory. We say it's involved in memory because we know that people with Alzheimer's disease seem to have acetylcholine producing neurons deteriorate, which means they don't produce as much acetylcholine. ACH, by the way, is the abbreviation for acetylcholine. Dopamine is another one that maybe you've heard of. This influences our movement, our learning, our attention, and our emotion. It can be quite a feel-good drug. In fact, cocaine works on these dopamine systems, which will explain how drugs work in a moment. However, again, in all your brain, it's a very delicate balance as far as the chemicals go. Too much of a chemical can cause a problem, not enough can cause a problem, so it needs to remain in a balance. So if there's an oversupply of dopamine, you have too much, we link that with schizophrenia. If there's an undersupply, we link that to Parkinson's disease. So not enough Parkinson's, too much schizophrenia. Serotonin, the one I gave you the example, of course, we link it with depression because it affects your mood, your hunger, your sleep, arousal, and it does a lot of things. Actually, it's involved in a lot of processes that we have. The antidepressant drugs are there to raise the serotonin levels or to keep it in the synapse a little bit longer. Norepinephrine helps control our alertness and arousal. It is also linked with depression and undersupply. So most of the drugs that we use as antidepressants work on norepinephrine and serotonin systems. GABA, or gamma-aminobutyric acid, is an inhibitory neurotransmitter. Now, excitatory neurotransmitters cause neurons to fire. Inhibitory ones slow down the firing, so they don't fire as much. So an undersupply of this is linked to seizures or tremors and insomnia. I'll come back to GABA a little bit later as well. Glutamate is also an excitatory neurotransmitter that's involved in memory. Now, an oversupply of this can overstimulate the brain and give you migraines or seizures, which is why some people avoid monosodium glutamate in food, the MSG. It gives them headaches, or in in larger cases or more severe, it could cause seizures. So a lot of people do avoid the MSG. This is not an exhaustive list of neurotransmitters. These are the main ones. We'll talk about some more in class. So what about these drugs? For example, we have drugs that are, a drug is anything that we ingest that works on this neural system. It's not a drug if it comes from our brain, even if it's not in balance. So it has to be something that we've ingested. Okay. Cocaine, LSD, ecstasy, crystal meth, any alcohol, all of those things. Some of them work as agonists and some of them work as antagonists. So we'll look at this chart here. Now, what an agonist does is copy that neurotransmitter. So the chemical makeup of the drug matches the shape of the neurotransmitter, because the receptor sites, again, work like a lock and key. The key has to fit the lock. So this is a representation of a natural neurotransmitter. Okay, say it's serotonin. So the serotonin neurotransmitter fits this lock perfectly. And once enough of them fit, then that neuron fires. So an agonist will look like it, close enough that it will fit that receptor site and make your brain believe it's actually that same neurotransmitter, even though it's a drug. So it causes those systems. So it's like an abundance of those neurotransmitters that you have. An antagonist, on the other hand, actually stops the receiving neuron from receiving the actual neurotransmitter. So these drugs will block, they'll fit so that they'll fit into the key space, for lack of a better description, but it doesn't fit good enough for your brain to think that it's the actual neurotransmitter. But it will stop the actual neurotransmitter from penetrating and getting into that receptor site. So for example, with acetylcholine, rattlesnake venom is like an agonist for acetylcholine. So it would be like your body has too much acetylcholine when you get bit by a rattlesnake. And what it does is cause muscle contractions and seizures. And this is why people will basically, you die from it because of the intense muscle contraction, convulsions. On the other side of the coin, there is a drug called curare, which is, I believe in South American tribes, sometimes they would dip an arrow into the drug. They would shoot an animal, an antelope or whatever it may be, and then the drug is passed into the system. So it's like a flood of these acetylcholine antagonists, which blocks those sites so the acetylcholine doesn't work. That animal becomes paralyzed and, of course, falls down. And the person goes over and thanks them and eats them. Thanks them because they made a good sacrifice for their family. Okay, we're going to leave it there for our first video. We'll come back with our second video, which is going to be about our nervous system and our endocrine system. Okay, so we'll see you guys in class. Goodbye for now.